All right, so this is the second part of the metabolism, and for the first part, we basically we visited the thermodynamics to better understand the metabolic pathways, how they're regulated in terms of enzymatic reactions, and like relationship between the KEQ and delta G naught, and also we visited some definitions of the critical terms for the thermodynamics, including what is it, the standard state, biochemical standard states, and so on. And we spent quite a bit of time to how the enzymatic reactions work and things like that. So today we're going to combine the what we've learned in the last lecture as well as a couple of previous lectures together to understand the metabolic pathway, especially for catabolism. So today we're gonna focus on the central pathway in the really complicated metabolic map or the pathways. So at the beginning of the last lecture, I've shown you a simplified version of the metabolic pathways. There are lots of substrates and the products were involved, the different kinds of macromolecules like DNA, RNA, lipids, and stuff like that. We're going to focus on the middle region that was highlighted in that map, which is glycolysis, and that actually forms the one of a central pathway component. Oh, recorded. I'm pretty sure I did it. I pressed on the record button. I don't know how to go back actually, but I did it. I'm pretty sure. So, and everything's unmuted, so it should be recorded in my voice and everything. One thing that actually reminds me that I wanted to make a quick announcement before the lecture that now you guys can fill up the SURF survey from the coming Monday, and if more than 8% of you guys finish the SURF, I'll give you extra 2 points to everybody. But it's 80%, so of the whole class, that's gonna be roughly, what is it? 205%, 164 students. More than at least 164 students completing that form, then you guys, all of you guys are getting 2 points, 2 extra points.
2 points on our last test or 2 points on our grade?
On top of the total grade. So it actually weighs quite a bit. And that, SURF survey is really important for improving my lectures and the content for the future classes, basically. Not you guys, unfortunately, but hopefully, it can be used as for the improving my lecture style and everything. So just to be frank, everything's anonymous, so you don't need to just talk about the good things at all. But I'll get all those feedback. Yeah, right. Given that we don't have 164 students here, so I'll do that. I'll make an announcement, basically writing an email or so. Alrighty, yeah, that was a good news that I should have told you before the lecture. Anyway, so let's go back to our lecture here about the central pathway. So that is basically for extracting energy from glucose. That's all about the glycolysis as well as some downstream pathways using the multi-step process. And the energy that is accumulated and extracted from this glucose will be transferred and accumulated in the easily useful form, which is ATP, as well as some other electron carriers, basically, as well as reduced coenzymes. Like, we talked about those coenzymes briefly, like NAD, FAD, and so on. So we're going to revisit them today too. So there are 3 components for this central pathway for metabolism. The first one is glycolysis, which is the top of those pathway that breaks down the glucose. And the second one is a citric acid cycle, also known as a Krebs cycle, TCA cycle, and so on. So these are basically the same thing. And lastly, electron transport chain. So During this, metabolism sections, we're gonna go over all these three key components of the metabolism, and we'll talk about all those steps and their regulations in much details. So today we're focusing on this glycolysis. Before you get to that, we want to talk a little bit more about high energy phosphate compounds, which is really critical for transferring electrons as well as extracting the energy and also save the energy in the sort of a transient storage form. So these compounds include ATP, phosphonyl pyruvate, 1,3-bisphosphoglycerate, thioesters, and so on. And commonly, they have all really highly negative delta G value. As you can see here, ATP that breaks down to ADP, and in inorganic phosphate has the delta G as negative 30.5. So it's highly negative, meaning that what does that mean? Like delta G value negative. Spontaneous process, so it can occur spontaneously, and that's going to be basically an exergonic process where this ATP is broken down to ATP and inorganic phosphate. Same thing applies to all these high energy phosphate compounds. Here ATP breaks down to AMP with the two inorganic phosphates. And here are the top two that I already talked about, here. So they are actually used quite often for again, reserving the energies or transferring energies. And also they play a critical role in glycolysis. Their hydrolysis should have delta G naught value smaller than negative 25 kilojoule per mole. So again, containing high energy or highly spontaneous process. And it's not for the long-term energy storage because it would have been just stored as a fat or the different kinds of carbohydrates. But they're only just transiently storing the energy in the form of this ATP or the other molecules here. And what they're doing is basically carrying the electrons or the energy from one enzyme system to the next system along this metabolic pathway. And it's called a high energy compound, not because it is unstable or the spontaneous decomposes. And it's pretty stable, especially ATP here. They are pretty stable molecules, but they are called high energy compounds because they can release a large quantity of the free energy upon hydrolysis. Again, these are the list and the delta G value, which is highly negative. And ATP especially directly participates in the reactions rather than it is simply hydrolyzed. So it can be actually, interact with the substrate of the enzyme through the metabolic pathway, giving the inorganic phosphate or phosphorus group to the substrate, or taking it off from a substrate, and so on. So again, phosphorus group or the nanolate moiety can be covalently attached to substrate or enzyme here. And that changes the energy state of those molecules, the substrate or enzyme, which will be used later. And basically it is activated, right? It took the phosphorus group, meaning that it contains the high amount of energy. So when there's a hydrolysis happens for this substrate or enzymes, it can basically use the energy for driving some other reactions. And it shows high energy phosphate compounds, low energy phosphate compounds, and so on. Again, these are often used for the metabolism, especially for catabolism. So here, now we're going to talk about the glycolysis. Again, this is one of the three critical components for the central pathway for metabolic metabolism, basically. And this is the first step, and it is central to the degradation of glucose. So it takes up the glucose, and it processes through these multiple steps, and eventually will generate two molecules of pyruvates. And this degradation of glucose occurs in the cytosol, meaning that all these substrates, products, as well as enzymes are soluble too, so they exist in cytoplasm. and they do not make the complex at all. So they're not associated in the complex or retained to the some kind of organelles or cell. They're just freely, diffusible. And this glycolysis process does not require oxygen. And that kind of refers us that it's probably was invented and evolved before there was a significant oxygen molecules built up in the atmosphere or accumulated in the atmosphere. And also this glycolysis process or the pathway is present in animals, plants, and microorganisms pretty much everywhere, meaning that this pathway is pretty much ancient. And the net result would be putting in the one glucose molecule to get 2 ATP. And again, ATP is a high energy compound, right? And it can be used as a temporary or the transient energy storage form. And role of glycolysis is to provide precursors for aerobic metabolism that happens after glycolysis that feeds into the TCA cycle, which is another component for the central pathway for metabolism. And the reason that is working in this like a separate way is that it makes it more specialized for certain molecule processing, and that makes this system more efficient rather than being lousy and little less specific in terms of their enzymes that are involved for processing those things, glucose, and breaking into small molecules. And again, it's used as the short-term energy supply when oxygen is limiting because it does not use oxygen, as it says here. It can just quickly generate the energy in terms of under the anaerobic conditions. And there are 10 reactions in glycolysis in total, from first one here to the 10th one here. And that occurs in 2 stages, and these 2 stages, each stage comprises 5 reactions, so 1 to 5 and 6 to 10. And this first stage, which is from step 1 to step 5, is the investment stage, where it actually spends 2 ATP. So instead of actually generating energies, it consumes energies. It uses 2 ATP for activating glucose. And activating meaning that it makes into the high energy state where phosphorus group can be attached to glucose, like glucose 6-phosphate. And again, 1 molecule of glucose is broken down to 2 molecules of glyceraldehyde 3-phosphate or GAP, which you can see right here. One's here and the other's here. So again, 1 glucose to generate 2 GAPs. And the second stage, which is from step 6 to step 10, is for paying off, basically. So now you yield 4 ATP. So in total, you lose or spend 2 ATP, but you yield 4 ATP. So in total, that's going to be 2 ATP that you're gaining from this glycolysis. And starts with the 2 molecules of GAP here again. And then eventually you're going to get 2 molecules of pyruvate, which is a simpler form here. Again, that yield is 2 molecules of ATP using just 1 molecule of glucose. And this pyruvate, which is the end product of glycolysis, may have one of these 3 fates. And these fates differ depending on the organism's cell types, conditions, developmental stages, and so on, many different things like while you're exercising or resting and stuff like that. It changes quite, quite quickly. One of those 3 fate is this pyruvate being oxidized in aerobes, which is animals and plants too, actually, when they're using the oxygens during night. And again, this end product pyruvate is oxidized, meaning that the pyruvate loses electrons, right? That's what oxidize or oxidation means. And eventually it will generate carbon dioxide and acetyl-CoA, coenzyme A, and this acetyl-coenzyme A enters to the next step of after the glycolysis for generating further energies, which is TCA cycle. And also, as another phase, the pyruvate can be reduced in lactate or lactic acid in contracting muscle. When you're doing a lot of exercise, you actually runs out of oxygen on your muscles and tissues and so on. So you can't really go through that aerobic process where this pathway is activated. So you get the lactate or lactic acids, and that's pretty toxic for you. That's why you're getting the muscle sore or the muscle cramping and things like that. And the last phase will be happening in yeast that is grown anaerobically, where there's a reduction happens of the pyruvate into ethanol and carbon dioxide through this acetaldehyde, which is a process of alcohol fermentation. So you can enjoy, being drunken basically using this product. So let's take a look at the energetic balance of one of those three phases, which is the glucose becoming the lactate or lactic acid. So it's basically breaking down the glucose into that 2 molecules of lactate. You can see the reaction here, and the delta G naught. Again, delta G is just a free energy of activation, not the activation, free energy of this reaction. But the delta G naught is the free energy at the standard state, right? And we also talked about delta G naught prime, which is for the delta G value at the biochemical standard state. But I typically just omit the prime there. So whenever I write down delta G naught, that actually means the biochemicals, delta G at the biochemical standard state. Okay, so this process has the delta G naught value with a highly negative value here, as over negative, like less than negative 183 kilojoule per mole. And this process is coupled with the gain of 2-ATP, where 2-ADP reacts to the 2 inorganic phosphate to generate 2-ATP and the 2 water molecule there, which requires energy. So delta G is a positive value, and that's what you want, right? During the process of glycolysis, after the glycolysis, what you want to do is generate energy. So what you want to get is these ATP molecules, but unfortunately, it requires energy, meaning that it's an endergonic process. But this process, breaking down of glucose, basically, is an exergonic process where it's spontaneously happening. So you can couple these two reactions to achieve these, generation of 2 ATP molecules. And when you think about the efficiency of these two coupled reactions, you're basically gaining 61 kilojoule per mole here, and your input basically is the here 183.6 kilojoule per mole. So if you get the ratio of that for the efficiency, that's about 33%. And that means that 33% of the total energy that is released during this process is captured for making the ATP molecules, 2 ATP molecules under standard conditions where delta G naught is used for calculating and telling the whether this is spontaneous or not. But in the cell, the synthesis of ATP costs more than 30.5 kilojoule per mole. It is actually 47.5 kilojoule per mole. How come there's a difference? I mean, this is a standard condition, but why is it different from this 30.5? Let's just consider that this is the biochemical standard state. Why, where these different come from? Anyone? Because I said that it's happening in the cell, and this value is basically at the standard state. Efficiency can be an issue, but let's not think about that at all. Let's say that this is just a one system that doesn't lose any energy there for biochemical reaction. It's a simple reaction. That's okay. I mean, I said it is at the standard state, meaning that the temperature should be 25 degrees C and the 1 atmosphere, and all the reactants and the products should be at 1 molar concentration. Is that what is really happening in the cell? It's not. So that's why, yeah, it can be. It is actually. Exactly. Biochemical as well as just the chemical too. It never happens in the cell. So you have to take into account actual concentration of glucose, ADP, ATP, and so on, right? So if you consider that, then you actually require more energy to generate ATP. So you're gonna see that a lot that delta G naught value is this, but actual delta G is different from delta G naught. So don't get confused. So making 2 ATP will require basically simply twice much of this, so which is about 95 kilojoules per mole. And if we use the real cellular concentration again, here's the formula or the equation that we used to get the delta G value. This is actually incorrect. So delta G would be delta G naught plus RT times ln and the ratio of the concentration of the products and reactants, right? And this delta G naught is pretty constant value. It's just a constant value because it's at the certain condition, and that's all. Same thing applies to R. It's just gas constant, and temperature we just assume that that's constant too as 37 degrees C. Then it's all about the ratio of the products and the reactants, right? So again, think about this relationship. And when you actually plug in the actual concentrations into this formula, then you can get 50% of efficiency, which is really high. Because the car engine efficiency is only 30% or less. So our living system is a highly efficient system to get the energy and transfer into the transport into the different form. All right. So I mean, many students have asked that what you should study, what you should focus. So I just actually make a list of that, the important things that you need to memorize or study here. So you basically need to know the actual reactions that are involved for the glycolysis with this, as well as how they are regulated too, and have to know the critical structures like a glucose 6-phosphate. They're not complicated because once you know the glucose, which is just a carbon ring with the 1 oxygen in the middle, that's a 6 ring structure. You can just get the 6-phosphate where you get the C6 and put the phosphoryl group there. Same thing applies to fructose 6-phosphate, fructose 1,6-bisphosphate, DHAP, GAP, and so on. So these are all the critical molecules that we're going to talk about during this lecture anyway. And all these basically that are the reactants and products in the glycolysis pathway, and pyruvate too. And you have to understand the 3 phases of a pyruvate that we just talked about, and also have to know the coenzymes that are involved in this glycolysis process. And these are basically NAD and FAD, as well as some other coenzymes here. So as long as you understand how they're involved and what their roles are during the boosting the reactions, should be good. And need to know the kind of reactions that these coenzymes mediate, like is it whether oxidation or reduction and so on. Again, once you understand how redox or the oxidation reduction reactions happens, then it's a really simple thing. Again, we're going to talk about this during this lecture. And you should know some vitamins that are also works as a coenzyme as well as precursors for coenzymes too. So need to know that. All right. So now we're gonna delve into glycolysis, and we'll talk about each step, where what are the reactants, what are the products, and how that reaction occurs, and what kind of enzymes are involved there, or the coenzymes are involved there. So the first step, which is on top here, is basically processing this glucose and activate it to make it into a high energy phosphate derivative, which is glucose 6-phosphate. And you can see their structures here too. And basically what it does is the phosphorylation at C6 of the glucose, and C6 is an exocyclic carbon of the glucose. So it's right on top of this ring, as you can see here. And that makes replace this hydrogen that is attached to hydroxyl here and replace it with the phosphoryl group here. And that uses 1 molecule of ATP per glucose. And this is basically the simple reaction here. Glucose ATP makes the glucose 6-phosphate and will be ADP. So this is the first investment step where you consume, where you use an ATP molecule, right? And that reaction is catalyzed by hexokinase. And we talked about the hexokinase pretty several times during we're talking about enzymes and as well as catalysis too, and also the first part of the metabolism. So you should be familiar with this hexokinase enzyme, which requires magnesium cation for its reaction. And how is it involved? I mean, why does this enzyme require magnesium cation? And that has to be coupled to ATP. So hexokinase's substrate is not just ATP, but it's ATP with magnesium cation. How? Why? Yes, it's a cofactor, but how is it involved in terms of boosting the reaction? Precisely. So the thing is that ATP is highly negatively charged, right? Because it has 3 phosphoryl groups attached there. And each phosphate group has 1 negative charge there. So it has to be stabilized for the electron negative attack, basically, to make this phosphoryl group attached to the glucose. So you're going to see that a lot, that whenever enzymes takes the negatively charged substrate molecule as a substrate, almost all the cases in the positively charged coenzyme is involved to stabilize that reaction inside the active site. So energetic balance for this reaction will be glucose that gets the inorganic phosphate to become the glucose 6-phosphate, which delta G naught value is 13.8. So it's positive value, meaning that it's endergonic. So it's not going to happen spontaneously. It's not favorable. But this reaction is coupled to ATP that is hydrolyzed into ADP, which again, spending energy, right? It's using energy to drive this reaction go forward. And the delta G naught value for this ATP hydrolysis is, of course, negative value here because this high energy compound. So this is the net reaction and the net delta G value. So again, this is still negative value. So it is a strongly exergonic overall. And meaning that it's going to keep going. So whenever this enzyme hexokinase sees the glucose, it'll accelerate this process, as well as even if in the absence of hexokinase, this phosphate will be turned to glucose 6-phosphate naturally. And this is not so great if you want to just hold that process and keep the glucose when you have enough energy there, right? So for this kind of highly spontaneous or exergonic processes, there are always some regulation steps involved there. So this is a good point where regulation can play and kind of make it as a sort of a checkpoint there. So how does that occur? So step 1, and it's regulation. Again, I talked to you that this is strongly exergonic with this negative value for delta G, meaning that ATP hydrolysis always happens and glucose will become glucose 6-phosphate. And again, under the cellular conditions, where the concentration of ATP, ADP, glucose are different than just 1 molar, then you can plug the actual number there to get the delta G, actual delta G, which is different from delta G not here, and that's even more spontaneous, right? Has a more negative free energy, meaning that it's gonna even go, more likely to go forward. And again, ATP hydrolysis, delta G naught value for ATP hydrolysis is -30, and it was considered as a highly spontaneous process, and that's why this molecule is called a high energy compound, right? So this is even smaller than, this delta G for this reaction is smaller than ATP hydrolysis, so it's gonna go really fast. So this one has to be regulated, this process has to be regulated, and that happens at the level of enzyme hexokinase. When you look at this hexokinase, phosphorylates the glucose, right? Generating the glucose 6-phosphate. And that means glucose will be charged now with a phosphate, negatively charged. And these charged molecules will not be able to cross the plasma membrane. So it'll just stay there. It is important to process to keep the glucose, which is the energy source, inside the cell. And also this hexokinase keeps the intracellular glucose level low because all the glucose will be become glucose 6-phosphate. So it promotes the diffusion of glucose from outside to inward, inside of the cell, using that concentration gradient. So whenever you take up the lot of glucose and that exists outside of the cell, you're gonna keep getting the glucose inside the cell using this process. And again, hexokinase, like I told you, have to be regulated because this is a highly spontaneous process. So it is actually acts as a control point, which is one of the 3 control points in glycolysis, where hexokinase is allosterically inhibited by its product glucose 6-phosphate. So we already talked about this actually in the last lecture. And this means that if there's a lot of consumption of glucose 6-phosphate using glycolysis, so it generates a lot of energies, it will lower the concentration of glucose 6-phosphate, and that will lead to the hexokinase that is reactivated and keep going this process for making more glucose 6-phosphate. Otherwise, if there's a lot of just the glucose, then it'll be inactive, right? Yes. So this hexokinase catalyzes this reaction up here, right? Making the glucose to glucose 6-phosphate. So that means glucose is consumed. Glucose level is going down as opposed to glucose 6-phosphate goes up. And their molecular properties are different, right? So since glucose level is going down, that's gonna keep the glucose level inside the cell low. So they're gonna, generally naturally induce the diffusion of glucose molecules outside of the cell into the cell. Okay. So here's another enzyme called glucokinase, and that is an isozyme or isoenzyme or alternative enzyme to hexokinase that we just talked about. And isozyme is basically like an enzyme in the different form. It can take the multiple molecular forms, but still use the same, still catalyze the same reaction. So hexokinase and glucokinases are isozymes, meaning that they have a similar molecular forms. different molecular forms, I'm sorry, different molecular forms, but still catalyze the same reaction where glucose becomes the glucose 6-phosphate. And these isozymes are often differ in kinetics and regulatory properties because their molecular forms are different. And they're different in identity of coenzyme use, localization, and so on. But they are related by amino acid sequences. So when you look at just amino acid sequences on their polypeptides, they're similar, especially at least for the certain domains that are critical for catalyzing such reactions. And that means they have actually similar structure too, at least for that particular domain that is responsible for this reaction. And that reflects the common evolutionary origin. So these isozymes could be possibly homologs, right? Because they can share a similar amino acid sequences. Are they homologs? They're homologs. So are they orthologs or are they paralogs? So we talked about that, a while ago, right? And why? Yeah, so that's correct. So they are paralog. Orthologs are basically sharing the same like a common ancestor, but there's a speciation where it changes to the different species, as opposed to paralogs are within the same organism or same species, but having multiple copies basically of the similar genes that are evolved in slightly different ways after gene duplication. So they are paralogs in many cases. So orthologs are only considered when you compare two species. But glucokinase and hexokinase can be found in the same species. If you want to talk about orthologs, you can focus on one hexokinase in mouse, in human, and in yeast, and can compare their gene sequences and so on. So you'd rather focus on just one gene, as opposed to these are two different genes that are transcribing two different enzymes, right? But happening within the same species. So that's why it's a paralog. I guess that's something I can ask in the exam. So this reaction that glucokinase drives, again, from glucose to glucose 6-phosphate occurs in liver. And its major role is to control glucose level in the blood, as opposed to hexokinase does it inside the cell, right? So it's about the glucose level inside the cell. So it's different inside the cell versus inside in the blood. And differences between this glucose kinase and the hexokinase is that the glucose kinase has higher Km value, and Km is a Michaelis constant for glucose. So it's 10 millimolar versus hexokinase's Km is 0.1 millimolar. So it's 100-fold difference there. And what does that mean? I mean, higher Km, is that a good thing for an enzyme or bad? Is it a good thing or a bad thing? Exactly. So Km tells you the affinity of an enzyme to its substrate, right? So that means glucokinase has lower affinity to glucose. So it'll eventually have a little lower efficiency too, right? So it requires more glucose concentration to be activated basically for catalyzing this reaction. So consider that the blood glucose concentration is 4 millimolar. It's not going to occur just naturally for this glucokinase being activated, as opposed to hexokinase will be activated pretty much, right? If that were existing in the bloodstream or in the liver. And this glucokinase is not inhibited by its product, so it's not allosterically inhibited by glucose 6-phosphate, but it's induced by insulin. So that's the difference from the hexokinase 2. The hexokinase doesn't care about insulin, of course, right? Because it's, again, happening inside the cell. And this glucokinase becomes metabolically important only when glucose concentration in liver is high. Again, we talked about this. Normal concentration will not do anything in terms of hexoglucokinase being binding the its substrate, but it happens only when this glucose level in liver is high, and this glucose level or the concentration in liver closely reflects the glucose in blood, concentration in blood. If you consume a lot of like sugars and things like that, your glucose level goes up in the blood, then it will actually reflect the glucose concentration in liver too, where the glucokinase will be activated to generate, catalyze this process from glucose to glucose 6-phosphate. And it will eventually will turn to glycogen in liver as a storage form. Yes. It can, but the thing is that they are happening in the totally different places. So the question was that what's the balance in terms of their activities between glucokinase and hexokinase? But again, hexokinase in the cytoplasm of the cell, and it's for the processing glucose inside the cell for glycolysis, as opposed to glucokinase exists in a tissue, especially in liver. It doesn't care about glycolysis. What it cares is that they are making into this storage form, glycogen. So they are involved in vastly different kind of biological process here, but they're still isozymes because what they're doing is essentially the same with the different kind of their properties. Okay, so let's move on to the step 2 here. So first, the step was basically activating glucose to make the glucose 6-phosphate. Now the second step is isomerization of this glucose 6-phosphate to fructose 6-phosphate. And as you can see here, this glucose 6-phosphate has the aldose form, as opposed to fructose 6-phosphate have ketose form. And this reaction is catalyzed by an enzyme called phosphoglucose isomerase, or PGI here. And this PGI enzyme is magnesium dependent. And as you can see, their substrates are basically, negatively charged because of this phosphor group. So it has to be stabilized by this magnesium using as a coenzyme here. And starting and final products are ring forms, as you can see, glucose and fructose, but the reaction occurs via linear form. So it's not going to be just breaking this up and bonding it as a sort of C5 ring here, but it's going to be complete linearization of glucose 6-phosphate first, and then process it back to forming into this. And it facilitates the subsequent phosphorylation of C1 here. So the major difference here is that this C1, which used to be endocyclic carbon, comes out of the this cycle or the ring and becomes an exocyclic carbon, right? And it is pretty good because it's outside of the ring, so it can be easily attacked by negatively charged functional groups, rather than included in the ring. So again, it just facilitates that attack at C1, and also facilitates the subsequent cleavage between C3 and C4, which is necessary to make it into 2 identical molecules in the later step. And this is an identical form, so it'll be easily attacked right here and make the breakage. But it has a small delta G knot value as negative 2.92 kilojoule per mole under the cellular conditions. So it's already near equilibrium, meaning that they can be freely reversible back and forth. And the next step right here is the making your fructose 6-phosphate to the fructose 1,6-bis phosphate. So that means it adds another phosphorus group here, especially at C1, to make the bisphosphate molecule. And again, it says there. And this is a second priming step. And priming is the same as activation, basically, where you invest some energies there to make the energies at the higher energy state. So later on, you can just use it easily to break it down to some other molecules without much of input of an energy. And here, another ATP molecule is consumed or invested. And this reaction is catalyzed by an enzyme called phosphofructokinase, or PFK. And also, this is driven forward due to the coupling mechanism here. This is some reaction, but when you look at the individual reactions, two reactions that are coupled here, fructose 6-phosphate gets another phosphate as C1 to get the fructose 1,6-bisphosphate, which have the positive delta G naught value. So it's not gonna, it would not happen spontaneously, but it's again coupled with the ATP hydrolysis, which gives a lot of energy. So the net delta G is negative. So again, this is a spontaneous process, favorable process. And in erythrocyte, which is red blood cell, delta G value is pretty similar, but lower. So there are important regulations happening on this step 3, where there's another phosphorylation happens at C1 of this fructose 6-phosphate. Essentially, this reaction is unidirectional, meaning that once it goes this way and that's it, cannot actually go back in most cases. And that means it commits to the metabolizing glucose from here, and once it happens, it cannot go back. It'll only go to the processing the glycolysis to generate energy, or it can choose go back to glucose and take another route for storing the sugars in the different forms, like glycogen. Since this is the committing step, it acts as a control point, so it's working as a sort of a valve for that glycolysis rate too. And this enzyme, phosphofructokinase, or PFK, is a tetramer, meaning that it has 4 subunits, and regulated allosterically in 3 ways here. Since this is a commitment step, there are a lot of activations and inhibitions. Basically, regulations are involved there to make sure that this is right, stage that it have to go forward. And then once it's go, then there are multiple ways to block it, from it coming back using the reverse reaction. So there are 3 ways here. The first one is using ATP, and when there's a high ATP concentration, inhibits the PFK activity. Again, this is the enzyme that is catalyzing this reaction. And that actually makes sense because when you have high ATP in the cell, you don't have, you don't need to generate any more ATP. There's already enough energy storage in the temporary form there. So it'll inhibit this activity of this enzyme to basically make the kind of log jam for the further processing this fructose 6-phosphate. When there's a low ATP, now it kind of works as a signal, go signal, right? So PFK, this enzyme will act as the Michaelis-Menten enzyme, which follows the Michaelis-Menten equation that we talked about in terms of its specificity, efficiency, and so on. And this one, this phosphofructokinase, which is a tetramer, and allosteric enzyme shows the sigmoidal substrate concentration curve. And an enzyme would normally have hyperbolic curve, but as you know, when it's allosterically regulated, it will show the sigmoidal, where it has that spatial property between the subunits within that enzyme complex, right? What is that spatial property? I'm actually looking for just one word for this, but why is it showing the like sigmoidal curve for rate of the reaction and substrate concentration? That means at the lower concentration, It's not going to have much of a binding and processing, but at the higher concentration, it'll accelerate it in terms of the enzymes binding to substrate and processing too. So it's more like a little like bimodal. How come? We talked about it, when we talk about any kinds of allosteric reactions, including hemoglobin. It's about cooperativity, right? They are cooperative in their subunits. So one's activated, and that actually induces the conformation or change of other subunits to change the modes for their binding. So we talked about sigmoidal. And we also talked about it in the last lecture. So if you don't understand, just go back to that. Also, this phosphofructokinase can be regulated by AMP, and this works as an allosteric activator as opposed to ATP works as an inhibitor. And that also totally makes sense. When ATP is used up, there's more AMP, and that means that the cell needs more energy storage stored. And the third regulation pathway will be using citrate, which is allosteric inhibitor, just like ATP. And this citrate or citric acid is an intermediate of a TCA cycle, which happens downstream of glycolysis. And that actually gives a signal that there are a lot of some intermediates that have to be processed in the downstream of glycolysis. So it gives the negative feedback, basically, that you have to stop or slow down. All right, so and that means TCA cycle, which is the next step of glycolysis, and the glycolysis are coupled through this PFK, phosphofructokinase regulation. The next step, step 4, is about cleavage of this fructose 1,6-bisphosphate into DHAP and GAP. And this reaction is catalyzed by fructose 1,6-bisphosphate aldolase, or just aldolase. And this cleavage happens between C3 and C4 of this fructose 1,6-bisphosphate. And it generates, again, DHAP and GAP. And the next step right here would be here. The DHAP is processed to the GAP, back to GAP, because only GAP can be used downstream of this glycosid pathway from the step 6 to the step 10. Again, this fructose 1,6-bisphosphate can generate one GAP molecule and one DHAP molecule here. But the only GAP can go further out to the downstream. So this DHAP changes to GAP using this enzyme triose phosphate isomerase, TPI or TIM, which is right here. And it gets essentially get the two GAP molecules instead of one GAP and one DHAP, right? Because DHAP changes to GAP molecule. And these two GAP molecules will have two identical legs, and they're going to fit into those two identical pathways later on. And this TPI or TIM, the isomerase for this DHAP to GAP change, it has the really high KCAT KM value. And we talked about KCAT and KM ratio, which tells us the efficiency of one molecule or the specificity. And already reaches to the catalytic perfection, meaning that its rate is similar to just the diffusion, 10 to the 8th millimolar. Okay, so far we talked about the first stage of the glycolysis, where it has the 5 steps, and that is an investment stage where it invested or used 2 molecules of ATP. And in the end, it generated one GAP and one DHAP. And again, this DHAP is changed to GAP. So it's going to be eventually have 2 GAP molecules there. And next stage From step 6 to step 10 will be the payoff stage where it will get the 4 ATP molecules. So since there are two legs, again, two pathways, and they are processing the same molecule, it means each leg will have 2 ATP generated. Okay, so we'll keep moving on these steps. Step 6, we're starting from this gap molecule, again, glyceraldehyde 3-phosphate, into the 1,3-bis phosphoglycerate, or 1,3-BPG. And you can, actually remember that on the list of high energy compounds, that was on top 1 or top 2. So it is high energy phosphate too. So you can imagine that this can be used further out in the next stage, where it can generate some ATP because it can hold a lot of energy inside this molecule. And the delta G naught for the hydrolysis in this 1,3-BPG is negative 49.6 kilojoule per mole. So it's highly negative again. It's enough, more than enough to generate ATP, right? Because ATP hydrolysis is with the delta G negative 30.5 kilojoule per mole. So the other way around of the hydrolysis of ATP synthesis will require 30.5 kilojoule per mole of energy at the standard state. And this is the first reaction in glycolysis that involves oxidation reduction. And again, oxidation reduction reaction is the where the coupled reaction is involved in transferring electrons. So we talked about this in the last lecture. And this aldehyde right here, which is glyceraldehyde 3-phosphate, has aldehyde on the top right here, is oxidized, meaning that it's losing its electrons to become carboxylic acid. So carboxylic acid is a COO minus, right? And then instead of hydrogen, it can have this phosphoryl group attached there. And again, quickly, oxidation is about the transferring electron from electron donor to electron acceptor. It's about losing an electron or multiple electrons. But it can lose it in the form of electrons or hydride ion too, which is hydrogen anion. And we talked a lot about hydrogen cations so far, right? But the same ion can be considered as a hydrogen anion too. Basically, it lacks one electron to fill the valency, right? So it can be either considered as having one extra electron, which is H plus, or the lacking one electron there, which is considered as H minus. So depending on its partner, you can consider it in either way. And since this electron is negatively charged, it's easy to think that this hydrogen ion is negatively charged too, as a hydride ion. So when you look at this reaction, it's not just happening by itself, but it actually requires a coenzyme NAD plus here, and it's positively charged, right? And it gets H here. So since this is positively charged, the partner that is getting into should be negatively charged. So that's why we're considering that as the hydride ion. But what is really happening is that this glyceraldehyde 3-phosphate molecule loses two electrons. One gets to this NAD to neutralize NAD, and the other one is getting neutralized the hydrogen ion that was detached from this aldehyde. So again, NADH accepts two electrons and it is reduced, right? As opposed to gap is oxidized. Make sense? And this process, this reaction is catalyzed by glyceraldehyde 3-phosphate dehydrogenase, or GAPDH. And it again requires NAD plus as its coenzyme. And hydride ion is transferred from aldehyde group to the NAD, basically here. And that's a redox reaction, oxidation reduction reaction. And you should understand what was oxidized, what was reduced here. And it basically energy is derived from there, from oxidation of aldehyde here, which generates a lot of energies. So it's a highly exergonic process, and that is used to form this carboxylic phosphoric anhydride component, which is a 1,3-BPG molecule, as well as drive the reduction of NAD plus too. And overall reaction, when you look at it here, which is the sum of these two delta G naught values, is positive, meaning that it's actually slightly androgonic. So it's not going to happen spontaneously too well, but it still happens because it can be coupled with the other reactions. So why does this androgonic reaction, which is critical for the oxidation of GAP, still happens even if it's androgonic? Because first, let's look at this reaction. Resulting high energy intermediate, which is this one, 1,3-BPG, is continuously used in the downstream of the reaction of this glycolysis. So we just talked about the step 5. Was it step 6? Step 6. But in the step 7, this 1,3-BPG is used as a substrate. So it's going to be continuously processed. So leads to the constant removal of this product. That means at the steady state, it keeps this concentration of 1,3-BPG at a low concentration, and that will still drive this reaction go forward. Because again, when you remember this equation for getting the delta G, which has this ratio of the concentration of product and reactants, if the product concentration goes down and stay low, then you're going to have negative values for this term, meaning that even if delta G naught is positive, you can still get delta G as a negative value. And that's what is happening in the cell, as delta G is actually a negative value here. And the arsenate is an interesting molecule that can substrate the hydrogen phosphate ion in this reaction. So here, the inorganic phosphate is used to be attached here to generate 1,3-BPG. And this can be replaced by this arsenate because its structure is quite similar to each other. And its reactivity too, because they are negatively charged. There are a lot of oxygens there. And if arsenate is replaced and involved in this reaction, it will generate this 1-arseno-3-phosphoglycerate, where everything is the same except for this arsenate instead of a phosphoryl group. And this molecule is unstable, so it will spontaneously hydrolyze into 3-phosphoglycerate, which is the product in the later stage of glycolysis. But the problem here is that there's no phosphoryl group attachment is involved there. So it's not going to get to the high energy state in terms of this making this 1,3-BPG. So basically the next step of the glycolysis is skipped. So since it does not reach to the high energy compound state, it will not lead to the synthesis of ATP molecule. So this is a pretty bad thing because oxidation of substrate, which is happening here, oxidation of GAP, is uncoupled from ATP synthesis, which will happen in the next step from this. And that's why this arsenate is a potent poison. That means even if you consume a lot of food and things like that, your cells will still stay starved because there's no ATP generated from there, right? Even if that glucose is still broken down. So the glycolysis becomes futile and no usable energy is extracted from glucose. So the next step would be this process where the 1,3-BPG becomes the 3-phosphoglycerate. So when you look at the structure of these molecules here, it's losing this phosphorus group from 1,3-BPG. And this is where this glycolysis breaks even in terms of its energetic gains, because this is the first step where it generates or synthesizes 2 molecules of ATP. So you already consumed the 2 ATP for the first stage of the glycolysis for the investment, right? Now you're gaining 2 ATP molecule here, so the net gain is 0. And here's the ADP to ATP. And these are 2 molecules again, because there are 2 legs of this downstream pathway of the glycolysis. And this is simply the transfer of this inorganic phosphate from 1,3-BPG to ATP. And this reaction is catalyzed by phosphoglycerate kinase, or PGK. And its true substrate is, again, not just simply ADP, but ADP with magnesium cations. And it's a highly exergonic process, as delta G is negative value, has negative value. And you can think of this 1,3-BPG as an intermediate between the step 6 and step 7. Again, step 6 here, we talked about that in the cell, it's barely spontaneous, right? So it needs another like process or the reaction that is coupled to make this reaction go forward more readily. So this step 7 is the coupled reaction for that. And the sum reaction here is the gap ADP gets to the 3-phosphoglycerate and ATP molecule in the involvement of this coenzyme NAD plus in step 6. And delta G would be just sum of step 6 and step 7, delta G from step 6 and step 7, which is a negative value. So again, it's gonna push the reaction go forward. And an example of this is an example of a substrate level phosphorylation. So there's a phosphorylation of ATP happens, right? And that happens at the expense of a substrate. So it takes up the inorganic phosphate from the substrate and just to put it to the ADP to make the ATP. And it's called the substrate level phosphorylation. And this is an example again, oxidation of GAP, which does not require oxygen, but there's another kind of a phosphorylation, which is respiration-linked phosphorylation, where it actually requires oxygen there. So it's going to be oxidative phosphorylation, meaning that it uses oxygen, and the substrate isn't involved there in many cases. The step 8, the next step, is converting the 3-PG to 2-PG here, 2-phosphoglycerate. And simply, it changes the position of this phosphorus group from C3 to C2. And this is the process of the preparation for making the next high energy phosphate, because this 2-PG is a better form, easier form to release the energy that you're going to see in step 9. And this is the reaction here, and with the delta G value a little positive, right? So again, it should be coupled with the other reaction that happens after this step 8 to again, push this reaction move forward. And it uses mutases for its catalysis, also called isomerases, and make basically making the isomers, right? So the mutases are the enzymes that catalyze the intermolecular transfer of the functional groups, like a phosphorus group, but can be hydroxyl group or some other groups too. And the mechanism here is that it's not again the direct intramolecular transfer, but it goes through this enzyme intermediates. So especially this mutase has an his-aided its residue in the active site that is phosphorylated, and this phosphorylated enzyme gets the 3PG as a substrate, and then give this phosphor group to substrate to make the ES complex here, where this BPG gets the 2-phosphor group, and then eventually, as a product, it will lose the 1-phosphor group that is attached to C3. And the step 9 is the dehydration of this 2PG, which is happening right here. And dehydration is taking out the water. So it takes out this hydrogen atom as well as hydroxyl group from 2PG to get the phosphoenol pyruvate or PEP. And this reaction is catalyzed by enzyme called enolase and also requires magnesium cation. Again, here's a phosphor group, so it has to be stabilized. And delta G is a slightly positive here too. And goal of this step is to rearrange the substrate in a way that it has the more potential energy eventually can be released upon the hydrolysis of this 2PG molecule. And delta G, when you look at the delta G for this, which is different from delta G naught again, is only negative 16.1. So it's a spontaneous process. So it can be coupled to the step 8, right? Again, driving the reaction to the one direction there. And again, I'm telling you delta G naught is different from delta G. Sorry about that. I should correct it. Delta G is different from delta G naught. And fluoride ions can inhibit the glycolysis because in the presence of inorganic phosphate, it forms this fluoride ion forms this fluorophosphate ion. And that means it gets rid of this inorganic phosphate, and that is involved for generating ATP out of from ADP. And it also this fluorophosphate forms the complex with positively charged magnesium cation in the active site of these enzymes. So instead of the magnesium cations involved in stabilizing substrate, it will just form the this ionic interactions with the fluorophosphate and will not do anything. So will make this enzyme inactive. Okay, so the last step, the step 10, is about generating another ATP by transferring this phosphor group from PEP to ADP. And that reaction is catalyzed by pyruvate kinase or PK, and it also generates the 2 molecules ATP, and it's a second step, right? So again, in total, from step 6 to step 10, which is the payoff stage, you get 4 ATP molecules in total. And that occurs via unstable anode tautomer. So this is this is an anode tautomer, and this is a keto tautomer, where you can see anode tautomer has a anode group, basically the alcohol group, which is O and something else, as opposed to keto group will have this ketone functional group. And the energetic balance for this reaction would be adding these 2 individual reactions. So PEP is gets the water molecule to get the pyruvate and loses a inorganic phosphate, and there's a ADP that gets the phosphor group to become ATP. So when you add these 2 delta G naught values, and the overall delta G naught is negative. So again, it can just go spontaneously, highly favorable reaction. And this is again highly exergonic and favorable because of this delta G naught value here. And you can plug this delta G naught value into the equation where we related KEQ and delta G naught, which was derived from this equation. Again, we talked about this in the last lecture, so you can always go back and check it. And when you plug this number here in the delta G naught, you can get KEQ as a 3.6 times 10 to the fifth power. It's a really high number, and KEQ is the ratio of the product and the concentration, right? So you have a lot of the products and less concentration. If you think it as a dissociation constant, if you can remember, in Michaelis-Menten equation, it's um, the concentration of enzymes and substrates over the concentration of ES complex, right? So they're going to be, instead of having some ES complex, you're going to have a lot of just enzymes and the substrates here. And that means equilibrium is shifted far to the right, so the forward reaction is strongly favored. So this is the condition where all those concentration of the substrate and the enzymes and the products is at equilibrium, right? But so it'll eventually push it that direction. Any questions here? Because it can be confusing, I understand that, but go back to the previous lecture and think about this again. And again, this has the highly negative delta G. So and that is due to the highly exergonic reactions that the NO tautomer becomes the keto tautomer or keto form there. And here's again, this is an NO form, and this is a keto form. And you can focus on this C2, where it has whether O something or the double bonding for O, making the keto form or ketone. And this keto form is more stable following the phosphorus group transfer there. And that's why this is a highly negative exergonic process. And also, since this is a highly exergonic, you want to put some regulatory steps there to use it as a control point, right? So once you remove the control point, then this reaction will go forward really readily. And then once you place the regulation, then it will stop there. So it can be pretty convenient to use another regulation point. So to do so, this enzyme which is a pyruvate kinase, is also regulated allosterically using this AMP and the fructose 1,6-bisphosphate 2, which both activates this enzyme activity. So and again, it makes sense because if there are a lot of AMP, that means cell needs more ATP to store the energies. So it'll just boost this process, activate the pyruvate kinase to process more glucose to generate ATP. Same thing applies to this fructose 1,6-bisphosphate, which was the product of step 4, I believe, if not step 3. Can be used as a glycolysis log jam signal. So when there's a lot of fructose 1,6-bisphosphate, then it has to be processed, right? So this it will activate the pyruvate kinase, as opposed to if there's a little of that, then there's not much of intermediates that should be processed further. So we'll just work as a sort of not activating much of this kinase enzyme. And this pyruvate kinase can be inhibited by ATP and acetyl-CoA. And this ATP totally makes sense because it's the opposite of AMP. So if you have a lot of ATP, there's no need to generate more energy. So it'll be inhibit this pyruvate kinase allosterically. And acetyl-CoA is the starting material of the TCA cycle, again, which is a downstream of this glycolysis. So if there's a lot of acetyl-CoA, meaning that there are a lot of stuff that needs to be processed further. So it will inhibit this reaction, the the pyruvate kinase from the driving this reaction going forward. Okay, so there are a lot of steps and the enzymes and the substrates were involved there, but this is a really critical metabolic pathway which is central. So you need to again memorize all these. Here's the overall equation that starts from glucose, and eventually you get the 2 pyruvate and 2 ATP here, and the electrons too that is taken by these electron carriers NAD plus. And here's the total reaction here, pathway here, as well as all the molecules involved. All right, so that's it.